All right, good evening, good morning, everybody. This is 8 to 12 watch, just coming on right now. So give us a few minutes to get set up.
You ready for,
Can we zoom out?
Yep, can do. Going wide.
All right.
Just in time for the geology lead watch. The rocks are looking really good here tonight.
They look like they stand right where they are, though.
Some of them, yeah. Some of them looks like they might come with us.
So,
We'll figure that out once we get things fully
problem
figured.
is apparently they picked up some huge rock and filled
Yeah.
up the starboard bio box, and like we're right at neutral now, so we don't have
Okay.
like huge payload.
What's our ballast situation looking like?
Well, they dumped one Alvin plate, so we have one Alvin plate, so,
Okay.
which is 16.5 pounds, which I'm already just at neutral.
Yeah.
So
Okay.
I'm supposed to be positively buoyant, so.
Right, yeah.
Ooh. That's a heavy rock.
It's a very big rock they picked up. That
It's
was
a little
dumb.
bigger than I usually like to get, but it was slim pickings for the first 4 hours.
Yeah.
If it's possible to pick up one smaller one at some point,
Yeah.
I would be more than
We're
thrilled.
just not going to be going on a grand rock hunt here. So.
Yeah, no. Let's keep Herc at least neutral, if not slightly positive.
Yeah. Yeah. Are we looking for anything in particular?
Not at the moment. I think we're still getting settled in. We've got a couple of seapens.
These umbellulas.
Yes, you got it. Umbellula. See, Robert listens.
Too muted. Too muted.
I love when the pilot knows what it is.
That is one of the seapen names that I know. So,
It's a fun one to say.
Umbellula. It sure is. All right, so what's our sample situation looking like? I know we're at neutral right now, so we can't take any more big rocks. So we might be able to get a small one, but
Are you looking for a rock?
She's always looking for a rock.
Yeah, Val's always looking for a rock.
okay.
We know you, Val.
We just got here.
So we recently collected the big rock. I take all responsibility for the ballast situation. So let's keep moving a little bit just to get things started and just see what we see. Get comfortable.
All right.
How does that sound?
It's great.
All right.
Super. I might be tempted to pitch that plate now, though, and
Okay.
make up for it with some thrust.
Okay.
We're supposed to be positive, and we're not.
Okay. Yeah. I'm happy if we keep it a positive environment. So if you want to dump a plate wherever you think is best to do so, let's do it.
All right.
Oh man, the umbellulas like it around here.
Yeah,
Yeah, they like this nice deep sediment.
Yeah, I think Steve was telling us in the lounge earlier, we usually find the seapens in about 10 to 15 centimeters of sediment.
Mm-hmm.
So that's a good indicator if you want to take a sediment sample, which we're not going to do right now. But that is something that we do have a request to collect during this expedition, or some sediments at some of our sites.
ROV team, do you think it's good for us to do some introductions right now?
Yeah, I think
Sure.
we're all right.
Yeah.
All
Go for
right.
it.
Well, welcome to anybody who is just tuning in now to Nautilus Live. My name is Giselle De Leon. I am a science communication fellow here with Nautilus Live. When I am not here on the vessel, I am a STEM educator in San Antonio, Texas. Anyone want to go next?
Okay, I am sitting right next to Giselle in the science lead position. My name is Val Finlayson. This is my third expedition with the Nautilus and OET. So I'm thrilled to be back. Was last on in 2023. I'm a geologist. I'm the geology lead on this expedition. My day job, when I'm not out to sea, which is most of the time, fortunately or unfortunately, I'm a high temperature isotope geochemist at the University of Maryland in the Department of Geology there.
Okay.
Want to go next?
My turn. I'm Luisa Dueñas. I'm from the National University of Colombia. I'm based in Bogota and also part of the science team as a biologist. Your turn.
Good evening, everyone. My name is Hovel Dako. I'm from the Solomons. I'm an intern here, an ocean science intern. As of right now, I'm a data logger, and I'm happy to be here. And everything else, we're ready to roll, and let's get it, shall we?
Yay, yeah.
All right, let's go.
In your front row, this is Megan Putz, your navigator. When I'm not here on the ship, I am a deep sea biologist at the University of Hawaii at Manoa.
Oh, sorry. I'm Robert Waters. I'm sitting in the Herc pilot seat. I am OET employee. I'm a facilities manager and ROV engineer when I'm not out here.
Yep, it's pressed down on SPL to make it white. And
Don't
then-
hold it. Don't hold it.
Yeah,
just
press
push it
and
down.
hold. Just press it. And then you go up here to listen. And now you're unmuted, so you can talk.
Hello, my name is Levi's. I am a mechanical engineering student at Columbia. I am the ROV engineering intern, and I am the Atalanta pilot. There it is.
And hello, I am Amber Flynn. I am the video engineer, and when I'm not on the boat, I am a cinematographer based in Los Angeles. I get all the cool buttons.
How fast are we going? 0.3?
We can do 0.3.
What are you doing?
We haven't moved yet.
Oh.
I wasn't sure if we wanted a rock here or not. It looks.
Did we want a rock here?
Let's hold off a little bit because we
Okay.
recently sampled the big one. So we'll try to get a little further up
Roger.
section. Yeah.
So we're going 250.
250 rock.
So it's the irony of ironies that I'm just sitting down in the lounge for the first third of this dive, just like, where are the rocks? Why can't everything's stuck? And now I'm just like, well, we're gonna wait a little bit longer now that they're all
Right.
accessible.
It's hard to sit back in the lounge and wait and watch everybody else do it.
It was more, I think, anxiety that the entire dive was going to be rocks that were firmly attached to the substrate, and I was gonna have nothing to work with because, like, this seamount would be really cool to have some geological information on. So we've got a little info on the seamounts a little bit further, well, a lot further north here along this little miniature chain in the Marshall Islands. So having data further to the south is extremely interesting for me and my team. So now that we have a rock on the vehicle, I'm much more relaxed about it.
A nice big one.
A little bigger than anticipated. I usually try to go for a little smaller, but at that point, I was in beggars can't be choosers mode. So we'll be a little more selective the rest of the dive for the heavy things.
Yeah. What would your perfect sample look like, Val?
Something with a lot of angular surfaces on it. Probably about 15 to 20 centimeters diameter, not 60 pounds. And yes, I'm mixing imperial and metric there.
Yeah.
Wait, is it really 60 pounds, the one that y'all picked up?
We
this
have
one we
picked up
don't
one
know.
that large before.
Hefty.
They are very dense rocks.
This one, we've picked up one similar in size to this before, and those are usually about 45 pounds, 20, 22 kilos. So
Nice.
it's going to be a big one.
You're
We're
gonna get
probably
your
going to
gym
want to keep it on the
workout.
floor in the lab. Yeah, get the kettlebell workout in with that. Without the handle, because we do things on hard mode.
Exactly.
And I know earlier in the lobby you were telling me about those angles. Can you elaborate more for anyone streaming in on why do we want
Can we look
angular
at this coral over here?
shapes on there? Oh,
Sure thing. So yeah, the angularity of the rock is kind of an important characteristic that we look for when we want to sample specifically lavas from a seamount. And that has a lot to do with the lava being really hot and fluid when it erupts, or mostly fluid. And as that cools,
Yeah, the yellower one.
it contracts.
One on the
And
right.
you develop structural weaknesses in the rock as it
Video,
kind of
can
pulls
we zoom
apart
in?
from itself.
Zooming.
And it will, as it breaks apart over time, it will preferentially break along those cooling fractures, those cooling joints. So you get some really characteristic angularity, like angular shapes, and some
Are we going
things
for
like
the
some
yellow
radial
coral? Okay.
fracture patterns that we look for. And that tells us, yeah, that's the kind of rock that we're looking for. So there are all sorts of like morphological and so like size characteristics that we try to aim for, and we can't always get there, but we do the best that we can. Especially with these old rocks, because we can't actually see what kind of rock we're picking up because everything's covered in this this knobbly botryoidal ferromanganese crust.
Close enough? You want to get closer?
I think that's good.
Ooh.
What is it?
This is a octocoral in the family Acanthogorgidae.
You got more zoom?
I got a little more.
Ooh.
That's
Oh,
full
wow.
zoom.
Little tentacle creeping out there.
Yeah, and on this coral we see a snake star. It is in the family Urrealidae.
Wow.
That's good for me. Thank you.
We're sort of out in front still.
Video coming wide.
Atlantis coming along. But we're still got plenty of time.
Another little sea
And then we
cucumber
have
up
that
there.
sea cucumber. That's a Paleopatides.
Ooh.
It's in the family Synallactidae.
So is one of its defining characteristics the very purple, I'm assuming, head?
Yeah, a lot of the synallactids tend to be purple or pink in color, but they can also be a little bit white. They
Okay.
generally have like a smoother body or a few protuberances along the body.
Okay, gotcha. Yeah, because there are
Where-
some that have, some other species that have just those really spectacular tentacles.
Yeah.
Yeah.
Yeah, so like the swimming ones, the Elpideids, they have a lot of those like little walking legs and what's called a velum, which is that sort of fin-like projection that they use to swim.
Cool.
That is so awesome.
We have a tiny glass sponge in the middle of the screen. That's a Bolosma.
Ooh.
Yeah, so one thing that was a little unexpected for us on this dive is how much sediment we've been seeing so consistently so far. And
So how old is these rocks here?
hard to say exactly, but they're somewhere in the probably 70 to 80 million year range. They're pretty old. And so we have a little evidence from further north. Those are 80 to about 87 million years old. So these are going to be slightly younger.
So can you tell by the crust when you get the rock up, or?
You can get a sense of it, but it's not a very consistent indicator, because usually the rocks that we're able to pick up, those tend to have broken off somewhat more recently, and they will have developed thinner crusts or had the crust broken off.
Right.
The ones that are intact that we're looking at right now probably have much thicker crusts that would be a somewhat better indicator of age. We can't really get absolute ages from those very easily, though.
Yeah.
Yeah.
Ooh, we have a little friend waving at us.
But yeah, I think we were expecting less sediment and more life on this ridge. So this one has been surprising, but every dive has its surprises.
Yeah.
That's the best part about this job.
Mm-hmm.
It's always so exciting.
It sure is.
Ooh, what is that?
Here we have a glass sponge in the family Roselidae. It's called Caulophacus.
Ooh.
You can identify this one based on where that long stalk attaches to the top of the sponge head. It looks a little bit like a mushroom, and that's how I know it's in the family Roselidae. Unlike the one we saw before, which attaches to like the base of the sponge head, and which is in the Bolosomanae.
Mm-hmm.
Video, can you zoom in?
Copy, zooming.
Oh, wow.
And some of these sponges can have associates on it. I see a little small white round thing.
tiny
It could
little
be a
one.
small little snail. Those seem to associate quite often with glass sponges.
Yeah, that little guy right there.
Yeah.
Yeah.
Yeah, those are really hard to collect because they are so tiny.
Yeah, I imagine I imagine they also are pretty attached, literally, to the sponge.
Yeah, you kind of have to collect the whole sponge,
Yeah.
and they're so fragile, and they don't have a lot of tissue.
That'd go right
I'll
through the
bet.
mesh in the jar if you.
Oh yeah,
Oh, yeah, that too.
exactly.
All right, moving on.
All right.
Coming wide.
I love this stream deck. So
Yeah.
fancy, so much power.
Damn
I
love.
can push so many buttons now. I'm so excited.
Oh no!
Press all the
Welcome
buttons.
to the joy of
Don't press them all at once.
Is there like a big red button that's really tempting?
The suction
no.
on button looks really nice to me.
Is there an on button?
Yeah, the suction on and start suctioning off.
Oh,
Ooh.
nice.
I'm kind of digging the new wide field camera.
Yeah, it's a great view.
So yeah, weather out here has been absolutely phenomenal today. We couldn't ask for better conditions for a dive. A little bit of a breeze earlier, but that seems to have calmed down. It's been warm, sunny all day.
Some few.
It was a
sharp
little hot on the deck
sightings.
for pre-dive setup, though.
Oh yeah, it was pretty spicy.
A little spicy.
All right, so in the corner over here, I'm operating the Multipuffer. I'm just, for the data loggers reference, I'm turning off pump 8 and 9, since we're at waypoint 9, and I'm going to turn on 10 and 11 for the next set of eDNA samples.
Okay, so which sample number is that?
It is going to be, so it's not in the same sequence as the Nautilus sample numbers, but it's going to be
okay.
MP10 and MP11.
Okay.
We can fill it in later.
Hey, can we reset Doppler when you get an opportunity?
So yeah, there you go.
No.
MP.
All right.
Ooh, I want to give a quick shout out to those who are just tuning in to Nautilus Live. I see we have some viewers from the United States, United Kingdom, Germany, Canada, Guam, Australia, New Zealand, Norway, and Italy. Welcome. If y'all are streaming on YouTube, I recommend you go visit Nautilus Live's website and join in the chat.
Sure.
It ran for, so 8 and 9 ran for 60 minutes and 50 seconds each.
Yeah, so this expedition, we are running a an in situ eDNA sampling setup. It's attached to the back of Herc, and we're controlling it to do very precise sampling on board the vehicle. And then once we bring the vehicles back up later tonight, we'll be processing those samples in the lab.
Ooh, for those viewers who might not
Give them
know.
a sec. I've got some ops.
Yep. So the for 8, number 8 was 165,928. And for number 9, it was 181,931.
Steve, how'd you get stuck running a micropuffer over there?
He totally volunteered.
Volunteer.
Wow.
Also, I wanted to attend the School of Rock. I need to learn how to rock.
I need to learn how to bio, so I'll be learning a little bit about sample preservation this cruise.
Can we zoom in video?
Copy, zooming.
Ooh, what are we looking at here?
So we've been seeing a few of these on this dive.
Yeah, so this is a Paragorgia. They're quite common at these depths. It's a type of octocoral, often called bubblegum coral. And on it, the yellow polyps are zoanthids, and they're parasitic on this coral. This one could possibly be in the genus Bologummi zoanthus.
Ooh.
And then again, we have a snake star, and we see this association quite often with these corals.
Mm-hmm.
Wow. Those colors are incredible.
Yeah, how to identify Paragorgia from other pink corals are the number of polyps you see at the ends of the branches. Paragorgia often has multiple polyps ending the branches, whereas hemicorallium and other corallids have
You can zoom out.
just paired polyps at the ends of the branches.
Gotcha.
Whoa.
I'll move ahead. Moving ahead.
9, it's 60 minutes, 48 seconds.
An Irridigorgia! A much more manageable sized one.
No kidding.
I have one request. Nav, can you open up those sample points for the last start of bottles 10 and 11?
Oh,
So
yeah.
I can get the lat long. All right.
By the way, what's the bearing to the next waypoint?
Next waypoint is... we're going to change to 245.
That isn't much of a change.
Not much of a change.
Just enough to stay on the ridge.
Yep.
Ooh, earlier we were talking about the micropuffer. Does anybody want to elaborate what eDNA is and why is it so important that we collect that?
Nav, can you open up that point one more time?
Oh, sorry, yes. This is some Acanthogorgia over here.
Yes, that's beautiful.
Yeah, zoom in on that thing.
Copy, zooming.
It's
And
a
it has
Chrysogorgia.
And it has an
Okay.
associate on it, another squat lobster.
Oh, wow.
Yeah, we often see this association of the squat lobster Europtychus with Chrysogorgia corals.
Oh.
And there's a tiny brittle star. It's so little.
Right.
From far away, I thought it might have been a different type of Chrysogorgid, just because it had a flat top, it could have been a
Looks like your tethers
Metallogorgia.
get a little low.
Right, yeah.
This one is definitely Chrysogorgia.
They do look a lot alike from a distance.
Oh yeah, it's hard
Yeah.
to tell.
You
You
gotta
have
keep
to look.
the delta, like, when I'm right under you, the tether will come up like it is, and just kind of come up a bit, just so you keep it from flopping up on top of Atalanta. So when I'm stretched out, you can come down lower, but when I'm right under you, you gotta come up.
All right, all set with that target. Yeah.
Okay.
We're gonna keep pumping on 10 and 11 until we get to waypoint 10.
Looks like another one of those bamboo skeletons just moving out of the left field of
I
view.
think that one might be alive. This yellow one.
Oh, maybe.
Ooh.
Yeah. There is a yellow bamboo called Irridigorgia
Can we zoom in?
S1 clade that is this
Zoom in?
color. The
There's
bamboo
something-
corals are notoriously difficult to work with and
So I've been
identify.
hearing. Yeah, Steve was saying a lot of the species tend to look alike, so visual identification can be really complicated. But yeah, this one is very yellow.
Yeah, these lemon yellow ones, we've just been calling S1 clade.
Okay.
We don't have a true identification yet for it, but there have been some samples. I'm sure some people are working on the genetics of these, but they're notoriously difficult.
All right, moving on.
Yeah, the
Zoom out.
S1
Wide?
clade typically corresponds, not always, but typically corresponds with the genus Cladorisis. But again, yeah, the genetics are in progress.
Gotcha.
Cladorisis is a very fragile bamboo coral species. It typically has very, very fine branches.
Is it kind of brittle?
It tends to be, yeah.
Okay.
Yeah. But we were actually, I was talking about the yellow coloration recently, and it's not something, it doesn't seem to be something innate to that clade, so it's got to be some other aspect
Something
of the
coming up.
biology.
Is that a... that's not a Walteria, is it?
No,
Oh, there's something else
it's
above it.
the one above it is Atlanticella, and that brown one, it probably was a euplectellid vase sponge that died.
Okay.
The Walterias are euplectellids, so it is a good guess.
Yeah.
But I would guess that was an old Regadrella or Dictyalis.
Yeah, I don't even know if we're in the range for Walterias.
Oh, we are. We definitely
Okay.
Did you want a closer look at it, or
Let's look at this Atlanticella. Those ones are really cool.
the white one?
The white one, yes.
I don't know if I've ever seen one of these before. These are cool.
Yeah, this one's really fun. It's basically a, you want me to go down?
Yeah.
I don't think I, there we go. There, it's
Just
slow.
kind of slow mo.
It's very slow. So the Atlanticella is a sort of a vase-like sponge. It has a single small attachment point, and as they get older, they kind of get more
Can you zoom in?
Copy, zooming.
abstract, you might
Oh,
say.
that's wild. They have some very interesting topology.
Mm-hmm.
Yeah.
It's a very soft sponge, so if you were to collect it, it'd be very soft and squishy.
Oh, wow.
Ooh.
That's an
sorry.
interesting feature for what I presume is a glass sponge, looking at it.
It is a glass sponge.
Yeah.
Yeah.
Oh, wow.
Oh,
Kind of
that's
reminds
gorgeous.
me of a fungus or something.
A little bit.
Yeah.
Yeah, sponges like this always fascinate me because the the structures that you can see are very mathematical, very fractal-like in a way. I just like stuff like that.
Up is a little
Yeah,
spicier than
it's
down.
beautiful.
Yeah. It's not great. We need an electric one
Yeah.
instead of a hydraulic. Are you happy with that?
I'm happy with that. Thank you.
Zoom
Round
out.
and wide.
Yeah, thanks for that. That was a really cool zoom. All right, so we are pretty close to waypoint 9. So we're moving toward waypoint 10. So we're cooking. So we planned a fairly long. Oh yeah. Oh. So we originally planned about a 1,200 meter track for this dive. 12 waypoints in all. We're
might be
coming
that
up on the last
Walteria.
couple here, so we may have to
Down
call an
the hole
audible and
there?
just kind of keep
Yeah,
heading up
you see
slope
this
as the dive
with
continues.
the crinoid on top?
Yeah.
Yeah, normally we move a little bit slower during these surveys, particularly when we're seeing a lot of different colonies and
Scoot over a little
a lot
bit.
of different species, but this one has been a little bit sparser, so we've been moving comparatively faster than we normally do.
Yeah.
So,
We zoom in video.
Tabby, zoom in.
so one thing we'll be looking for as we ascend higher is kind of looking for a transition potentially between the volcanic substrate and when we might start transitioning into the carbonate cap if we get that high.
Ooh.
Yeah.
There's your Walteria, Val.
Oh, there we go.
wow, look at this.
That crinoid is quite a bit larger than the Walteria.
Oh, yeah.
But any sort of height advantage getting off the substrate helps with the feeding.
Yep.
Yeah.
And
Get
it-
into that water
We gotta get
column.
moving again.
Yep.
Zooming out.
Coming wide.
There was a ctenophore on one of those branches on the Walteria too.
Oh, I totally missed that. Good spot.
Hidden, but yeah, brown, mucusy looking.
I need to get better at spotting ctenophores. I have not been very good at that.
Something very tall in the back there.
Should we go check it out? You can see some animal traces on the substrate as well. It's those dark lineations that we're seeing here and there. Some of them are cracks, but some of those are traces left behind by some sort of shelled animal, like a limpet or something.
Oh, wow.
Bamboo.
It's a nice thick bamboo.
Yeah. It's pretty long.
It just keeps going.
It's like the Energizer Bunny.
Yeah. Oh, wow.
Ah, is that better? Can you move your mic a little bit closer? Should be about a finger away. Yeah.
Better?
Cool. You need zooms or you happy?
I'm happy with that.
Yeah, I think that's better, Amber. Thank you.
Hang on.
Wow.
All right.
Okay, one sec. right, so we're seeing more ripple marks here in the sediment, and they're all running pretty parallel to each other, so that gives you an idea that there is a sustained current that's running in one direction along this seamount. And similarly, we can also see some striations sometimes in the ferromanganese crusts coating the rocks. And when you see striations like that, that's also controlled by current. That's a little bit of an erosional feature. Here you don't see that, so you see more of those bumps. That's what we call botryoidal texture, and it kind of looks like bubbles or like a bunch of grapes, and some hydrous minerals, so some stuff that precipitates out of like settings like this, out of water, can have that crystal morphology sometimes.
Oh, wow.
Yeah.
What do you want?
Nothing.
Oh, okay. You looked at me. I was like, okay.
Looked like you were going for the controls.
Oh, I like the controls.
Yeah.
But
That's
we've
Stream
seen enough
Deck, right?
Umbellulas.
Yeah, press the buttons.
The power.
Keep Megan away from that suction on button.
But it's so fun.
It's orange. It's like really enticing.
That's a really good color for button pressing.
Isn't that usually a warning color in the animal kingdom?
It is.
The most enticing one is the red button.
Is it cherry flavored?
Or strawberry.
Another tall bamboo.
Yeah.
Ooh. Somebody earlier said that they want to use it as a back scratcher. How good would that feel? I don't know.
Maybe, but I don't know. Some of these animals, they do smell a little interesting when they get up. I don't know if I could handle that.
Right?
Maybe a little stinky.
That is a large bamboo. Another one.
Wow.
This one doesn't look
Yeah.
as lively
Coming off
though.
This
middle
one's
there.
got some squiggly bits in the middle.
Squiggly bits.
That is a great descriptor.
I love squigglies.
And sometimes when these bamboos get damaged, they can start growing branches, and some branch naturally. And this one looks a little strange.
Wanna zoom in?
Copy, zooming.
I usually call something like this a sparse brancher, sort of making it up as it goes.
You think somebody tried to munch on it for a while?
It
At
could
some
be, you know, sometimes they get a little damage and then it just like starts branching at that spot.
Okay.
Yeah, you can see where it's branching. There's an anemone there.
Yeah, I see the anemone.
Oh.
Yeah, right there.
Uh-huh, there it is.
What kind of animal would predate on a bamboo coral?
A lot of sea stars actually eat bamboo corals.
We may
And we'll
see some
probably
of that
see
at some
that.
point.
Yeah, there's a lot of corallivory on bamboo corals.
You see a lot of pink sea stars, basically Patrick, and those tend to be pretty predatory.
Yeah, the cookie stars are very predatory on these corals.
Okay.
Yeah, so those are like the Evoplosoma and the Hipasteria.
Do the cookie stars look like cookies?
They kind of do.
They kind of
yeah.
do. Yeah, some of them are more cookie than others.
Ooh.
I'm actually surprised we haven't seen any.
Oh, I want to see a chocolate chip one.
They usually come in like white and orange.
Ooh, those are good colors too.
Yeah, they've got some really interesting patterns on, I guess, the top-facing portion of their bodies. Don't think I've seen the underside of one.
Okay. Carrying on.
Commonoid. Nice flying. That was beautiful.
some more sponges, and we're getting back into a segment of rock that looks like everything's pretty crusted down, kind of stuck again. We've been in and out of patches of loose and encrusted rock.
Is
So-
there a sponge there?
Yeah, it's a dead one.
Dead sponge.
It's got a crinoid on it.
How can you
But
tell
there's
when
a little
sponge?
sea star right at the base there,
Yeah.
down to the left. Look at that. That's new for
Oh
our
yeah,
dive.
this one. Go ahead.
Yeah, right there.
Ooh.
So it looks like most of the
Can you
vase
zoom in?
sponges that we've seen have been dead,
Copy, zooming.
with the exception of that Walteria. So if a lot of the vase sponges haven't been happy around here, do you think they might be getting choked off eventually by the sedimentation?
I don't think so. I think they
Okay.
just get old.
Just get old?
Yeah.
Yeah, don't we all eventually?
Try to figure.
You're lucky.
You're lucky, yeah.
Can we take a look at this guy coming in the center here? On the bottom?
I'm done to see. Oh, I see.
I think it's a bamboo. But it might be a primnoid.
You push in on that one?
Ready for a zoom?
Looks a little primnoidy.
Yes,
Yeah,
Ever zoom?
I think so.
okay. Look at that, the rock person getting a little better at biology. You learn a lot running watches on this ship.
So it's definitely a
Not a-
primnoid.
Not a good angle.
Cool.
Do you
Yeah,
wanna
we're
go
looking at it
in
kind
a
of
little
edge
more?
on.
Do we have more?
You want to look at it? You want to go around?
No, I can see it fine from here.
All right.
I just want to see the polyps.
Oh.
Yeah, that's good. Definitely a primnoid, probably Norella. Thank you.
Common white.
Got a Bracingid sea star. We've been seeing a lot of those. They're kind of fun. They're filter feeders, so when you see them with their arms up, that's their feeding posture.
And they've been quite large around here.
Oh yeah,
Like-
they can get quite big.
Yeah, we've seen what? Some around 30, 40 centimeters, maybe even bigger.
Mm-hmm.
What's it doing out on the plains there and not hanging out on a coral?
They don't usually like to be on corals. You'll see some on corals, but yeah, these ones almost always are on rocks. They also really like shipwrecks.
Oh, interesting.
Yeah,
So they like the harder substrates
they like
then.
harder substrates that don't move around.
I don't blame them.
Zoom in again.
Copy, zoom in.
Got another really tall bamboo off to the side.
Yeah,
we
here's
can
another
see
one
it.
of those.
We can see it in the wide
primnoids.
field.
This one's a nice one.
That is. That's a shrimp on the crinoid. Okay.
Oh yeah, tiny little Caridius shrimp.
Mm-hmm. I love watching them swim around. They just look very stately, like they're just off to go do some shrimp business.
Yeah, shrimp business.
kind of business do shrimps have?
They have a lot
I
of
don't know.
business.
Yeah, they look like they're off to do something very important when they're swimming around.
They really do. They look so busy.
It might be something like Candidella, this one. The polyps are very skinny. They don't have that three-tiered look of the Neurala.
Yeah, they're very delicate looking on this one. I don't know if we've seen one of these yet this dive.
We can zoom out when you're ready.
Coming wide.
Hang on.
Gorgeous.
That's a nice shot of the whole colony.
Oh, wow, yeah.
More life happening now.
Mm-hmm.
Are we getting near the top or something? Where are we at?
Top somewhere around 1,300 meters on this seamount, if I remember correctly. So we still have a ways to go.
That sounds.
Wow. But the sediment is a little bit less here, steeper slopes, so there just might be more places for animals to hold on to.
Oh, there's a cup coral.
Oh yeah, I thought I was seeing a couple of those earlier, but I wasn't sure.
Are you looking?
Yeah, so right at the bottom, there's a little
right at the
pink
bottom.
thing.
I see.
Okay, I wasn't sure if I was looking at some of those deep sea barnacles or cup corals earlier.
Wait.
That potentially
see it?
answers that question.
Is it
Yeah,
hidden?
it's like right
hidden.
behind the
Yeah,
coral.
it's hidden. Yeah, let's
There it
scoot over.
is.
Oh, that's a big one.
Peekaboo.
No, that is not what I was seeing earlier.
All right. Want to zoom in?
Yeah, let's
Copy.
get a quick zoom on that.
Oh, how cool.
It's quite the juxtaposition to the primnoid with the size of the polyps.
Whoa.
And you can see the plates. So this is a
Yeah.
hard coral, so it has those little plates. It has a very skinny little base.
Yeah.
So it could be
the
something
size of
like
that.
Calomyces. But the cup corals are hard. There's a lot of them.
Is the cup coral like one big polyp, or
Yeah,
is
it's
each
just a single
plate-
polyp.
Oh,
I think this is the biggest one I've ever seen on one of these dives.
Yeah, they
Are we
don't.
supposed
Yeah,
to be over there?
yeah, we're supposed to be over there.
Right. Pick it up.
Coming wide.
Cool.
There's a cool sponge over there.
Mm-hmm. We're definitely
wanna
seeing
look
more
at
in
it,
this
or-
area.
Sure, we can look at it. Unless it's too much in the cabbage.
The what?
That's what Gabby always tells me. Like, I get lost in the cabbages.
Instead of lost in the sauce.
Yeah, lost in the sauce, lost in the cabbages.
As long as we don't get too far off ridge, yeah.
This is a Regadrella. It's another glass sponge in the family Euplectellidae.
So I have some hydroids on it?
I don't think so.
Okay.
But it usually has a shrimp in the center. So if you look down the barrel there's usually a pair of shrimps that live in there. And they're called Spongicola.
Oops, scared 'em away.
Spongicola.
I saw them in there, and then they.
Yeah,
scared.
it saw us.
Got
Uh-oh.
shy.
Oh.
So like, is it a breeding pair that tend to live in these sponges?
Yep, that's right.
Okay.
Yeah, once they go in as little babies, they can't go out. They just live in their studio apartment for the
So-
rest of their lives.
So this
mean,
is
they've got
the
quite a
wedding
view.
sponge.
The wedding sponge, yeah.
Oh.
So in Japan, this has been known to be given as a wedding gift.
Oh.
Wow.
Do you want to take a zoom on this one, or?
Sure, if you want to look down the barrel there, you might be able to see.
There is something kind of pink-orange in there.
Pinkish, yeah.
I definitely saw it as we were approaching, but then it zoomed away inside the sponge.
Ooh.
That is so cool. The center
There
kind
it
of looks
is.
like
Yeah.
spider webs.
There's something inside.
Oh.
Oh, there you see it.
Oh.
Yes, it just
Do you
moved.
have more zoom?
I can zoom when Robert's ready.
Okay.
You got more zoom?
I haven't zoomed yet. I'm waiting for you to...
Oh, yeah, go
I'm
for
good?
it.
Okay, going.
Enhance image.
Enhancing.
Maximum zoom.
Oh,
There we are.
There it is.
Hey there,
See
buddy.
its little shrimply eye?
Yeah.
Oh, wow.
Yeah, they just hang out in there.
That is so cool.
So what happens if there's more than two?
I don't know, but this one is a female, and it has eggs.
Wow.
Yeah, you see her eggs and her pleopods?
Is that the male over there on
Is
the,
it this?
no.
on the left?
That-
A little up a little bit?
Yeah,
and
must
then
be. The
just
other
went out
one's
of
the
field
male.
of view. Yeah, the other shrimp is there, correct?
Oh, that is a polychaete.
Oh, it's a polychaete. Okay.
Yeah.
Oh, what's a polychaete?
Roommate.
It's a kind of worm.
Oh. More
Maybe it's the pet.
tenants.
Yeah, that's our pet polychaete. It's like having a dog, you know?
Yeah, exactly. Yeah.
I wonder
Alright.
what kind of noise it make.
That was
Carrying
cool.
on. Carrying on.
Now we can get back on track.
That was a very good side quest.
It was.
I really like those Regadrella sponges. They're always really fun.
I agree.
I think that was the best look of at one of those Spongicola shrimps that we've had.
Yeah.
Especially to see the gravid female.
Hello, sea stars.
Oh, wow. Yeah.
Yeah, all
That
those
must be a
brittle
really
stars.
good rock to hang out on.
They like it right there.
Yeah, it looks like a great place to sit if you ask me. I'd sit with them.
They do like their ledges.
They do.
Alright, since we're at waypoint 10, I'm going to stop pumps 10 and 11.
We're
Okay.
at the waypoint 10. We got
Yep.
like 20 meters.
We're not really going through the waypoints, kind of paralleling them for most of the dive. Are we planning on going northwards?
Like to
Nah.
it. I was gonna just zip over there so I could call
Okay.
it a done deal.
Yeah, I think we're aiming to get back on top of the ridge.
Okay.
And then I'm just going to keep planning on heading uphill once we get past waypoint
Yeah,
12.
we keep going on that heading, we drop off the ridge right there. Unless it's just a local thing.
Either way, I'm going to stop pumps 10 and 11 now and hold off on starting 12 and 13 for a moment.
Okay.
Very lumpy in here.
Yep, super lumpy.
Ooh. It's an Irridigorgia.
Oh, let's. Oh, no,
It's dead.
I think there might be a long arm spiny on there.
Is there?
I don't know. I was looking at the Cinemacam.
Yeah, it's not looking very lively at all, is it?
Oh.
Oh, yeah, there we go.
What were you looking
So,
for?
like, right above those two yellow crinoids, I think there's a squat lobster.
Wanna zoom in?
Right here, right?
Yeah.
No.
Copy
The
zooming.
squat lobster.
No, there's one on the tip?
There's a big
No, it's a.
snake star or something.
It's just a brittle star.
Yeah.
But it was doing the posture with the arms. It got me.
Yeah, it's just hanging out like a squat lobster. Maybe it's
Tricked me.
Halloween down here.
All right.
Okay, on.
Okay,
Yes.
coming wide.
Yeah, they're stopped at yeah. Hold on. The time stopped was 0901.
Went too far. There you go.
UTC. And I am gonna start
Close enough.
the next
Waypoint
two.
10.
Yeah. All right.
What's
90603.
Can you go back to the public chat?
Yep, 33:55. 33 minutes 55 seconds. For 9 or 11? 11. 33:54 minutes. 95629. Yeah. And I will start the next ones momentarily.
All right, so we've got some interesting questions coming in on the public chat. I'm busy peeking over Giselle's shoulder here. And we've got somebody asking about the manganese crusts in this area and how they differ from the cobalt crusts near the Mariana Trench. And the cobalt crusts and the ferromanganese crusts here are actually quite similar. There are some different zones throughout the Pacific where the exact metal content within these ferromanganese crusts differs a little bit, and some are of more economic interest than others because of some of the different quantities of the metals contained. We don't actually know much about the ferromanganese crusts in this particular spot that we're diving in. Some of that material is going to go back to the archive for some researchers to work on, but especially up in the marine sanctuary that we'll be working in later in this expedition, those are off limits for any sort of economic work, regardless of what their content is, because we want to try to preserve the seabed in these areas as much as possible, particularly given, we are working in the waters of an island nation, and water is, that's the lifeblood of this kind of area. But, yeah.
I have a thing I want to look at.
Ooh. Okay.
You see that little pink thing, Robert? There's a little pink guy.
Oh, where?
All right, I'm gonna start 12 and 13 now, MP 13 and 12.
I think it's a polychelid, a type of blind lobster.
Ooh.
All right, 12 and 13, pumping.
Did you call for a zoom, Robert? Okay, I can't hear you very well.
Nav, can you drop a target for start 12 and 13 pump?
Yeah, this is a deep sea blind lobster
Oh, wow.
Look at you.
called Homerian asper.
That is amazing.
They have these really crazy long arms. They're really fun. Yeah, he's got them tucked in.
Ah.
How cool.
And then there's a little shrimp, Caridian shrimp, some kind up there.
So if this is a blind lobster, how does it navigate usually?
Well, it can navigate well, it's so dark down here, it doesn't need eyes.
Yeah. Oh.
So it can navigate based on its other senses of smell and touch. But I thought you guys would appreciate this one. It's one of my favorite strange and wacky sea creatures.
It's adorable.
It is.
Nav, can you drop a target for MP 1213 start?
Covered what?
Yep.
I think you're muted, Robert.
I think so.
Yeah. I was
Okay, thank you.
talking to Levi.
Copy.
No worries.
And can you...
Do you want that up open?
Yeah, just for a second.
Yeah.
Alrighty, thought I'd welcome some viewers who are just tuning in
Okay.
right now.
That's good.
Cool.
If y'all want to leave a comment, ask us any questions, you can go to nautiluslive.org. Operations, do you think we're okay for another introduction for anyone just tuning in?
Looks like we're gonna drop off the ridge that way.
Are we?
Yeah. Maybe it's just local, but
Yeah. I'm
We
just
see-
following.
sonar says it's just local.
Yeah.
All right. Carrying on.
Carrying on.
Sounds good. Yeah, once we're kind of closer to the ridge crest, I might be interested in trying to look for another rock, if we see anything loose. So it looks like the water's getting a little bit less clear here. There's a little bit more
Well, we're
of a
dropping
sediment
off
and
the edge,
organics
though,
load.
so
Okay.
Yeah. But I'm seeing some more marine snow in the cameras.
Oh
I'm kind of going down slope, so you're gonna have to follow me, Levi.
Yeah, I think we're moving through a small saddle point here,
Yeah,
so
it
I think
looks
it is a
like-
local feature.
It's a local feature, I think.
Yeah.
What's this thing?
Still a lot of sediment up here.
Is this another stock sponge? Yeah.
Yep, it's a Caliphacus. But it might have a shrimp on it. I think I heard there might be interest in associations. You see the little pink on the back side, that little dot there?
Okay.
So-
I can't go too far out in front here because
Yeah, it looks like there's a shrimp in there.
Atlantis gonna get dragged on the rocks. All right, you on the camera?
Yep.
Video zoom.
Zooming.
So we have a Levius type of curted shrimp on this Caliphacus sponge.
Is this something that we want to sample for associates work?
There's two of them, actually.
Yeah, there's
one
two of
the
them on this
backside.
one.
If it's a good spot, I think it might be worth a suction.
Okay.
How much move we got?
We're going 30 meters, but I could have us.
Well, no, we need a little more. 'Cause
I know. Yeah, we're coming.
I think we'll be okay.
Okay.
Okay. We're good for sampling?
Yeah.
Okay.
Let me get more situated here.
Okay, no problem.
Coming wide.
So that's interesting. There, you see that kink in the stalk on the
Yeah.
sponge? Do you think it's a sponge that grew on another stalk, or do you think something might have happened to the stalk itself?
I'm pretty sure like something happened to the sponge and it branched off. And some of these Caliphacus sponges can actually have multiple heads.
Ooh.
I
That's
don't know.
cool.
Yeah, I've seen the most heads I think we've counted was like 10.
10?
Wow.
I think
Yeah.
I've seen like 2.
All right, I'm kind of getting yanked around still. We're gonna have to wait a second.
Yeah.
Okay, no worries. But yeah, the vast majority of the time you see the Caulophacus sponges with just one head.
Yeah, the shrimps are gonna want to swim away, so we'll have to be quick.
Yeah, I figured that might be a one-shot suction attempt.
Mm-hmm.
They do tend to move quickly when they want to.
Kind of side slope here.
And I get to touch the suction button.
Oh, yeah. We need a porch out.
Perch out.
Achievement about to be unlocked.
Yes.
Yes. The forbidden.
Yeah.
Seat out. Arms up.
Right, which sample are we on? 14?
I have 20.
Okay. Yeah,
Yeah,
my bad.
We gotta look at the jars. Okay, Levi, you gotta learn about cameras. So we need to switch to bucket. So you gotta turn that one off, turn that one on. And then we're on, what bucket number are we on? Can you see?
It looks like 2.
What do we want to be on?
Data logger, do you know what sample jars are open?
Stand by. We're locating that.
Oops. Why is the jaw so snappy here?
It's spicy.
It's way too much. Huh. That's better.
So all of them are full except for number 1 cellars.
So 2 is good?
All of them are open except
Okay.
for number 1.
So we're currently on 2, so that should be good.
Yeah.
Oh, it's creeping too. It's got the creeps.
All right.
Okay, so
There are 2 there.
Yeah.
can we zoom in?
Copy, zoom in.
Zoom way in because I'm not going to be able to tell very good here.
A little more? Or
I think that's okay. Give it the beans.
Do we have more beans on the section?
Are you sucking?
I'm sucking. Suction's on.
You gotta hold it down.
Oh, I gotta hold it?
Yeah, you gotta hold it.
All right.
You holding?
I'm holding. But the little particles in the tube are not moving around.
Not at all? What's that?
Should I turn it off?
They had this issue earlier. They just had to cycle the jars.
You had to move the jars?
Yeah, just cycle the jar back
Oh,
and
that's
forward.
strange.
Okay, let off the suction.
Okay, suction off.
Okay, so Rotate, dang it. There she goes. Okay, you all over it? Okay, we got it on.
Okay, it's on.
Uh,
Is it on?
Ooh.
Oop, oop.
Dang it.
I don't see the particles moving in
Oh.
there.
Ah! Zoom out!
Oh no.
I can't see him. Wait, oh, he's on the basket.
Got away.
Ah.
No.
No.
All right. Back over here. We still sucking?
I don't think it is. Normally the little particles in the jar are moving, but I have it on. Let me turn it off on mine.
Yeah.
You turn
Okay,
it
they're
on.
moving now.
Maybe my button doesn't work.
Oh, that's disappointing.
Is it going now?
Oh no.
Oh, wait. Oh.
Is it? I don't see any action.
Yeah.
I don't see any action
I don't
either.
see any.
Oh, that's weird.
Why? Why no action? It's going,
Okay, it's going.
Okay.
Back on.
but shrimp's on the hiding behind the backside here.
Yeah, it's tricky.
Oh.
I'm like very
Ah.
close to a wall.
Yeah.
So
Hang
pull
on.
up. The winch. I don't think.
No,
it's,
there's no
it's not.
suction.
There's no suction.
Dang it.
Otherwise, it would go in.
Whoa, what's
I'll get-
happening there?
You're hitting the
Look,
ground.
Come
up,
You're going
come up.
down. Oh my God. You set it down on the ground.
Oh,
Oh my God.
Okay. We're back up now.
sorry.
Good thing it's rugged. Not cool.
Okay, well, we'll look for another opportunity.
Yeah.
I think we may want to try to move on top of the ridge and see what that looks like. I know Fanny was interested in trying to
Right.
get a
Get back on it.
transect.
All right, so we're abandoning this?
I think
Yeah.
so, yeah.
Yes.
Yeah, shrimp are hard to get.
I don't know what's up with this slurp there.
Yeah, we should
Yeah.
check that out.
Yeah, let's go see if we can try for transect for Fanny on top of the ridge.
Okay.
Because if we are in kind of a saddle point, it's going to be a little bit flatter around here, and that we'll see if it's suitable for her.
Suck in the drawer.
Porch in.
Porch. Yeah, that was bad.
Well, we have identified that something's up with the suction, so that's a good thing, because then we can fix it.
All right.
There.
Is sonar okay? Yeah.
Yeah. So we're just gonna head up a little bit,
No
and
harm,
then
no foul. Suction off.
so the transect we want to do 50 meters.
Yeah. I just heard from Fanny. She will let us know if it's suitable.
Okay.
What are we doing?
They want to do a video transect where we're gonna keep same altitude and no zooms for 50 meters.
But it's very
Yeah, we're just gonna
lumpy.
see.
Yeah,
think this
we're
is
gonna
good.
try.
Whether
We're
it's
gonna try
suitable.
to keep.
Like, we got to do it right here? 'Cause this
No.
is not a good spot. This is like the
Yeah,
lumpiest we've seen.
Let's
No,
go
but
to like the top of the ridge.
yeah, let's follow the top of the ridge for a little bit and see if there's something that looks
So,
suitable.
Is this? Would we say this is the top, Robert?
This
Or
looks like the
one-
top.
Okay.
Yeah. Yeah, we might be slightly offset from the map.
Okay.
Okay, you gotta get more up here.
$290. Okay. I'm gonna call
So
and move.
this is the most important thing. Never mind all that other crap. Just keep on this. Drop it. Just don't even look at it.
Alright.
Just pay attention to that.
Okay.
I don't care about any of that. I care about that a lot.
Yeah. Altitude. Very important. A cheetah gnat. There we go.
What?
It swam away. It was a little wiggly thing.
Very skinny ridge tops here.
It is very skinny.
So what is this transit thing we're trying to do?
So they want to do a video transect that's 50 meters long where we don't stop and zoom, and we keep an even altitude above bottom as possible, probably between 1 and 2 meters if possible.
Okay.
The issue I'm seeing is that there are not a ton of corals to have in the
No.
imagery.
this
No, there's
A
nothing.
coral survey.
Coral
Yeah.
survey, yeah. Not too many. It's a little sparse.
So are we going to try and do it, or what do we think?
Let's keep moving upslope. So
Okay.
let's keep on our approach to waypoint 11.
Okay.
Yeah. Unfortunately, just
250?
Yeah, I made a 280 move laterally because I thought that we were going to try to follow this feature.
Okay.
But yeah, we're going to start heading down to 11.
Okay.
if
So my
possible,
next move, I'll move down.
okay, I was gonna say if possible, could we stay on the ridge?
Okay, do you want to just follow
Well,
the
it
feature?
sorta-
Yeah, let's follow the feature.
It looks like that's pretty
It's
much.
kind of the direction it's
Yeah.
going.
Yeah, I
I
think
think
we
that
just have a little.
locally it looked like it was going a little bit straighter
Yeah,
to
we're
the...
just there's some wiggliness
there.
to it, but
Yeah, we're seeing more detail than we can capture in the map resolution. So I'm more inclined to follow the feature at this point.
okay.
Yeah, I mean,
I think
we're within error.
We're good. It's following the right heading.
Yeah, we'll
We just
adjust
got
as
local
needed.
stuff. Just some local bumps.
Lumps and bumps. It's that creative process of geology.
Yes. A little bit meandering, but it gets the job done.
Mm-hmm.
Note for the multipuffer that pump 13 is not pumping any volume, but 12 is looking okay, so I'm guessing it's a pump failure on 13. I'm
What do
going
we
to turn
got
it off.
going here? Okay, you got auto
Thanks, Steve.
heading off. That's no good. What's happening here?
Pump 13.
Hang on. Oh my God. Why is that not going on?
That's weird.
Alrighty, just wanted to do another shout out to all of our followers who are tuning in right now. We have people from the United States, Guam, the United Kingdom, Germany, Canada, Australia, Taiwan, Philippines, and Norway. Again, for anyone tuning in on YouTube, I recommend you go to nautiluslive.org to ask us any questions. Is anyone good for any reintroductions for any new people tuning in?
Sure.
Yeah. Well, my name is Giselle. I guess I'll get started off. When I'm not on Nautilus Live as a science communication fellow, I usually work at a science museum in San Antonio, Texas.
All right, Val Finlayson, I'm the watch lead. I'm a geologist and the geology lead on the ship this expedition.
All right, something, no.
Stand by.
What's going on here?
All right, yeah, ROV is working something out here. So I might be a little start-stop, but yeah, my day job, I'm an isotope geochemist. I work on a lot of the rocks that we recover out here. Back at the lab at the University of Maryland in College Park.
North.
Luisa Dueñas from the National University in Colombia. I'm based in Bogota. I'm here also with Ocean Census. I'm a biologist and I'm also part of the science team.
Good evening, ladies and gentlemen. My name is Dept. I'm a data logger here. I'm an intern here. I'm from the Solomons, and I'm a bachelor student attending the University.
The heading's not changing. I don't have heading.
Thank you.
I don't have heading.
Yeah, and front row is working on some stuff, so we can ask them for introductions later, but in the meantime, we'll let them handle what they need to.
All right, well, I got to stretch out here because I don't know what's going on. So. Well, I got to stretch out because I don't want Atlantis to spin around. We don't have a heading. It's just, it's saying it's always pointed the same way.
Got our first fish. This is a halosaur.
Oh, wow.
All right. Thank you.
Yeah, heading's not working.
That's kind of need that.
Yeah.
All right. On the Multipuffer, I'm going to stop, since we're close to 11, I'm going to stop pump 12 and 13 right now. Call those samples complete. And I'm ready for volume data whenever data's ready.
Can I please have the minutes run for pump 12?
12 is 16 minutes, 13 seconds.
Total milliliter of water?
45,000.
All right, well, the
49.
heading's just not working.
49,049.
Yeah, 45049.
Thank you. Now for pump 13 minutes run.
15 minutes.
15 minutes. Total milliliter of water?
22379.
Thank you.
I made a note that after about 9 minutes, it stopped pumping, so it may be blocked, but I think there's only about 9 minutes of good pumping on that sample, but I made a note of it.
All right. Thank you.
Yeah, we have no heading. No heading.
Did we lose the compass on Herc?
No, we lost on Atalanta,
Oh,
No.
okay.
which,
We've never had an Atalanta heading.
what?
We haven't had Atalanta heading in a while.
What are you talking about?
Oh, it usually does not.
No, Atalanta has to have auto head.
Oh,
It
okay.
has to be able to hold heading. You have to know what its heading is. Most definitely we have.
Well,
heading.
sometimes, well, I guess you've had it on your thing, but this symbol doesn't
Well,
always change.
that got fixed. Though.
Oh, that got fixed?
Yeah, so it has been working. It's currently, we don't have any heading. See,
At all.
I have nothing.
That's not good.
Yeah.
So that's why I didn't see it as a problem, because my screen didn't always have heading.
It's definitely a problem.
It's definitely a problem when your screen doesn't have it.
Yeah, we have to have heading. So
Are we calling it?
I'm gonna have to call it because I have to have heading. I
Okay.
can't not have heading.
Yeah, if we can't
We
navigate,
can get
then
away with stuff, but not that stuff.
okay.
And yeah, the coincidence of the lack of heading and what happened kind of makes me.
Yeah, nervous
Yes.
that something might have happened.
I'm thinking the
Understandable.
same thing.
Yeah.
Okay.
So I would like to recover.
Okay. I agree.
All right, I am going to run the last two multipuffer samples on the ascent. 14 and 15, I'm going to start them now. At 9:35.
Yeah. We have no idea how many wraps we have. There's many things we don't have.
We need it, yeah.
Okay. Yeah, I don't want to fly blind.
We can't.
No.
We're not gonna.
Nope.
No can do. All right. I'm gonna start coming up.
All right.
So your job is to keep the delta at close to 0.
Yeah.
Can we still take some Niskin samples on the way up?
Sure.
All right, let's go ahead and pop bottle number 3.
So in case of sampling, how do you know where to put the...
You just choose.
Or you just choose which one?
So, Data, you ready for taking a sample for Niskin number 3?
Okay, has this got camera rack back on it somewhere?
Yes, retract.
Retract.
Number 3.
Is it already retracted?
Must be.
All All right, when
Okay,
Bob
can you follow
pops
me over?
Yep.
the ring for bottle number 3, we'll log that as a sample.
Yeah.
Oh yeah, we did. So now this is sample 25.
It's not 23
Stop
and
that.
24.
Yeah, and they're about to take 25. And then there's
Are we watching? Did we already trip it?
I'm watching. Go ahead.
Did it go?
I didn't. Okay, I see. 1, 2, 3. I think it went.
Yeah.
Is it upside down?
It's upside down this year.
Okay.
I know, it threw me
I'm
off
so
too.
confused.
We're looking at the bottom of the
Yeah,
bottles.
I see.
So that's definitely, yeah.
Yeah,
So there's
it's
4 of
close,
them tripped because
close.
you can't see the first
First one.
So starting from the back
Yeah,
here.
I see 3
I'll do
closed.
three ropes hanging down there, and the one you can't see because it's already too far back.
It's over here. Okay.
Yeah.
Changing it up on me.
I know, it threw me off too.
We did that last cruise.
I was trying to show my video team where all the cameras are. I'm like, I cannot find the Niskin one. Now I know.
I don't know if it's better or worse, but it is what it is.
Which one do I write on?
All right, can we center this business up here?
Yep.
Have to write another one down here. Oh, what? 22. So the grub is using the hands. Slurp is the pipe. Slurp grab is both the hand and the... And then the Niskin, what's the Niskin?
That's the bottles on the side. I'll show it to you when the ROV comes up. Actually, I might have a photo.
Oh, okay.
So they just capture water, 5 liters of water.
And then push forward.
So the compass lives inside the bottle, so it's not something that's hanging out there that could get whacked. But it took a bump.
Back here,
Yeah, so they grab it and then just push it into
Yeah.
the sediment. And when they pull it out, it has like a core full of mud.
Okay. Scoop.
Yeah.
Alright. Major.
Major, we don't use that on this.
Alright, I got it. Let's go.
Yeah. We're coming up at
Fill out the forms
we're coming up at
4.
3 and 24.
Alright. Okay.
19, 20, somewhere. 20 meters a minute. 19. 95 minutes. Ish.
Alrighty, for anyone just tuning in, we are currently working towards collecting water samples in something called Niskin bottles. Can anyone tell me why we're collecting these water samples?
We'll give the biology team a sec to log some stuff, and
yes,
then they'll
yes.
answer the eDNA question. Unfortunately, I cannot do that justice.
I feel I can't either.
Yeah.
All right, I have some guidance on the remaining 2 bottles. I think we're gonna pop 1 bottle approximately, well, let's see. This deep scattering layer would have changed. Let's do, let's do 600 and 200.
600 and 200.
600 and 200. So 600 will be 2, bottle 2, and 200 will be bottle 1.
Rog.
So the last sample, what was the sample number?
I don't have that data. What was the last sample number for the Niskin we just took off bottom?
25.
25?
25.
25.
Okay. Look at this little floaty jelly over by the
Oh, I
tether.
totally missed it.
Little baby.
Yeah, every now and again you'll see during dives these really small jellies, and they are often pretty strikingly colored, and they're totally predatory and will munch on corals.
Oh, wow.
Yeah.
Oh, that is so cool. I'd love to see that
Usually
in
like
action.
kind of this bright red color.
Ooh.
They're
Yeah.
very small.
We see them on the Irridigorgia quite often.
So they have a preference.
That's what I've noticed.
Interesting.
Sometimes
Okay.
bamboos too.
Yeah, I've seen them on bamboos too.
Ooh, so they got a favorite.
They have a name, and I don't think I'm authorized to say it yet, but the paper is making its way through the process.
Oh, that's
Yeah.
exciting.
Yeah, it's a new genus and species.
Oh,
Really?
fantastic.
That's exciting. And you're working on that?
I am not working on that. My superiors are, and I will convey that as soon as I'm able to.
Awesome.
Ooh, exciting.
But, yeah, I think we have well over a dozen specimens from all over the Central Pacific, so it's a really neat one.
You know, it's actually kind of, well, maybe I shouldn't be surprised because I know this is how science works, but it seems surprising to me that, you know, this is an animal that we do see pretty frequently on dives that is only just now kind of getting its place in the animal kingdom. But there's just so much work to do in the deep sea, and not a ton of us doing it.
Most taxonomy is done by people who are not actually paid to do that specific work.
Yeah.
It's
Yeah.
That's right.
side projects on side projects, or you know, done during a sabbatical or something. You know, it's It's just one of those things.
Which is interesting because it seems like that should be an entire branch that should just be like a thriving full-time science.
You're
Yeah.
preaching to the choir here.
Oh yeah. Well, as the rock taxonomy equivalent kind of person, yeah, we have the same problem. There's not a ton of us doing the kind of work that I do in geology, so.
Is that a thing, rock taxonomy?
I was just about to ask that.
Not formally. We call our jargon for it is isotopic fingerprinting. But you know, we're doing sort of a similar thing, looking for distinctive markers in some different isotope systems. So that's sort of our version of genetics. It's probably a little bit simpler in some ways.
Sure.
1048,
Yeah.
1150.
All right.
Right? Yeah, 1150. Right? Is that what you got? 1150? Expected time on surface?
Yeah.
Well, that works out okay.
Yeah.
We got another watch coming on. Surprise, surprise.
Surprise. Well, we'll have enough people for deck.
Yeah.
That was the concern when we do the watch change in the midnight, like between watches. It's hard to find people.
The 2 a.m.? Yeah.
Yeah, 2 a.m. is a really hard time to find people.
Yeah. It's about the hardest.
In the meanwhile, Nav, can you drop a target approximately on MP 1213 and call it MP 1415 start?
Okay.
And leave it open so I can copy some coordinates.
Oh. Come here. I dropped it too close. No.
I can hear you adjusting your mic.
So Val, once we get the rock, what are you going to do with it?
Once
What's
we get
the process?
So there are a few steps on board, many more back at the lab, but the initial processing is working with the science managers. Once we get the vehicles back on board, we recover the samples off the vehicle, and there's an order of operations for that. So the really sensitive water and bio samples go first, and then we'll get the rocks. There will be some initial descriptions and photography that take place, and then if there are any associates on the rocks that that the bio team wants to sample, they're going to recover those, and after that, then I start working on the rocks. So I won't be doing this until tomorrow when we have daylight, just for safety reasons, but I will, you know, once I've confirmed that everybody has everything that they need for the initial documentation and biological work from the rocks, I'll take those over to the saw in the morning and start cutting those open.
Okay.
So that way I can get a look at the texture of the igneous rock or whatever we pull up, and at that point I can start doing what's called a petrographic description. So I look at things like what minerals are in the rock, vesicles, I get sizes, masses,
Oh, he's not coming out.
more photos of the interior once it's cut open, and I just try to get as detailed a field report as possible.
I guess
Great.
Yeah,
That sounds interesting.
yeah.
I want to see how the saw works.
Oh yeah,
That'll be
you'll see
fun.
plenty of that. Yeah. We'll also do some subsampling because I'm going to send some small or relatively small pieces of these rocks to my home institution so that I can work on them in my lab. I've got a project that is going to be working on rocks from this part of the Pacific, and we're really interested in the geologic history of this area and what sources of volcanism helped contribute to the building of the Marshall Islands, and get an idea of what things looked like in the Pacific Ocean 70 to 85 million years ago. And I'm also going to send off subsamples to one of my collaborators who can determine the ages of the rocks. So I kind of figure out where they come from in the mantle. He figures out what the ages are. So we work really closely together on this kind of work. And then most of the sample is going to go back to the Geological Repository at University of Rhode Island, where any researcher can request pieces of those rocks to do other things with. So we make these accessible to the community.
Oh, that's great.
It's such a good resource. And we have multiple geological archives throughout the US and all over the planet, really.
Nice.
So there's
That's
some
good
really
to know.
wonderful collections out there.
Great.
Yeah. But yeah, I will be happy to show you the workings of the saw and what I look for in the rocks.
Yeah, for sure.
So, yeah, we've got one on this dive. We'll be working on it tomorrow.
Yeah, great.
Yeah, and also am I working on samples during the day instead of in the middle of the night. It's not just for safety reasons. It also keeps me out of the way of the biologists who need to do their job more quickly, because
Yes.
the samples that all of you are working on are much more sensitive than mine will be. Mine can wait a few hours. They've been down there for tens of millions of years. They can wait. They can
A
wait
little bit more.
longer. Yeah.
Val, question for you. If there's associates on your rock, will they first be kind of processed and looked at by biology peeps first? Okay.
Yeah, I'm gonna let the biology team go over the rocks, and yeah, if they find any associates, they'll sample those. So that's something that we haven't done a ton of in the past, and there are a lot of smaller animals, smaller corals, other organisms, sometimes little miniature sea stars that are attached to the rocks, and it would be really interesting to get better documentation of those. You know, they're often overlooked.
All set with that target nav.
You hear? He said he's all set with the target. Steve.
Yep.
Steve is set with the target.
Sorry, Steve. Cool, thanks. Wait, did it not change my name? No.
Oh, you're fitzing around now. What are the options? Something?
Louisa, you're getting a shout out in public chat for Ocean Census.
Yay. Yeah. Thank you
mind
for that.
Yeah.
Let's go.
Would you mind telling us a little bit about, could you tell us a little bit about Ocean Census and its mission?
Does it work?
Yeah, sure. Ocean Census is an organization that looks to advanced species discoveries in the deep sea. Because we know that there's a lot of different species down there, but we haven't actually described them yet. So through Ocean Census, the idea is to help with that discovery of the species. And that's why I'm here as part of the science team as a taxonomist to try to look at the different octocorals maybe, that's my
How about
specialty,
Awesome.
and see if maybe they're new species, if they're different or not, and just start the process of trying to identify them.
Wow. Any octocorals that you really want to focus on or that you're really interested in seeing on this expedition?
Well, I'm really interested in primnoids, but also not only octocorals, but still sterid corals. Those are hydrocorals.
Okay.
So that